Hello children, today again I'm here with molecular basis of inheritance. Today we will go to the 9th lesson where we discuss about mutation. Now first and foremost we have to know what is mutation. Mutations are sudden inheritable changes which occurs in the genetic material. Now as we are already familiar, the genetic material can be either RNA or DNA. Most of the cases it is DNA, deoxyribonucleic acid, acting as the genetic material. Now if a change occurs in one or more of the base pairs in DNA, that might result into a mutation. That might be called as a mutation provided the expression of the particular those base sequences changes from the previously whatever they were expressing as. Those such type of changes in base sequences, if they are inherited, that means they are passed on to the next generation, they are carried over to the next generation, then it can be called as a mutation. That is, the expression of the character due to those base sequences changes compared to the previously existing ones. Such changes are called as mutations. Mutations can again be of different types. The main types here, as we can see in the slide, there is mutation can be basically of two types: frame shift mutation and point mutation. In point mutation, there is if a particular base pair, let us say AT, that was the base pair at a point within the DNA. Now that gets replaced by, let us say, GC, which was previously AT. It got changed, or it got substituted, it got replaced by another pair GC. Accordingly, there will be a change in the expression of those the expression of the bases present within it. So such type of mutations can be referred to as point mutation or substitution mutation. The other type is frame shift mutation. In this case, what happens? One or more of base pairs may be added to the already existing base pairs, or maybe they are deleted from the already existing ones. So which will result in total shift of the triplet codons. There will be shift, okay, which we call as frame shift. We will be discussing about both these two types in details. Now first and foremost, we also have to know what causes mutations. Two main reasons have been cited as the reason for the occurrence of mutations: spontaneous and induced. Spontaneous, as the term suggests, it occurs on its own. That is, DNA can undergo chemical changes, okay, it undergoes the change spontaneously. Maybe during the transcription process it undergoes some changes. During replication it undergoes changes. That is, where a base a thymine had to be added up, what happened? A thymine did not add up, some other bases replaced it. So it can occur spontaneously. The exact reason not yet may be deciphered. So there occurs. So replication errors may occur. So one in, say, millions, in one replication, such a change, such a mutation is likely to occur. Now the DNA polymerase which proofreads new strands, okay, during that proofreading, during that reading of by the DNA polymerase, you have to relate to replication to know about it a little bit. So during that time maybe some changes occurs, okay, but usually there is correction of errors. But sometimes, which if the error is not corrected, then it may result into a mutation. Induced mutation is of more common occurrence, that is, which is due to some agents referred to as mutagens. Mutagens are agents that causes mutation. The agents may be in the form of radiations, it may be some chemicals, okay, and some mutagens are also carcinogens, cancer causing. So what happens if we are exposed to those certain agents, they are likely to change the base sequences and accordingly change the expression of the total character or nature in the individual. Then there are some also environmental mutagens in the form of UV radiations, tobacco smoke. These are all some environmental mutagens. Now again, a few mutagens we will discuss here. Causes of mutation: physical mutagen, chemical mutagen, or biological mutagen. Physical mutagens can be in the form of radiations like X-rays, UV rays from the sun. Okay, so here UV or X-rays may be the reason for causing mutation, causing changes in the base sequences. There can be some chemical mutagens in the form of pollutants, which we are always, most of the time we are exposed, tobacco smoke, some pesticides, insecticides, chemicals. Here there's a illustration also. The chemicals can be some carcinogens like the cigarette smoke, tobacco smoke, processed foods which contain preservatives. Those are chemicals, and they may when for a long duration, for a long time we are constantly consuming it, it may ultimately affect our genes, affect the DNA sequences. Cosmetics and cleansing products also affect. Perfumes, deo sprays, even they affect in the long term, they may have some deleterious effect in the form of mutations. Then the biological mutagens also exist, like some viruses, human papilloma virus. Okay, this is a human papilloma virus, okay, which if infects the person can bring about changes in the base sequences. Even some bacteria, Helicobacter is a type which can also bring about mutation in humans. So these are some major causes of mutation, induced mutation, which we call as mutagens: physical, chemical, and biological. Now we will go to the types. First is point mutation. As I already mentioned, point mutation is change in a single base pair. Only a single base pair changes, and that is it is replaced. That is called as substitution mutation or point mutation. Like say for example, here as we are already familiar, we have learnt genetic code, we are already familiar that base sequences, they are read in triplets. The codons are read in triplets. So here we are not taking the base sequences, but just we have taken a sentence here, okay, each word of which is formed out of 3 words. Let us say each letter here represents one base. Okay, each letter represents one base. So like the first three letters will mean one codon, next 3 letters will mean another codon, and so on. The, T-H-E, the, it makes sense. The, it forms a word. Dog bit the cat. This, let us assume it to be the normal DNA sequence, which is definitely making some sense. It makes some sense. If in its place we take some DNA sequences, okay, DNA sequences like say A, U, G, that makes sense in the form that it will code for amino acid. So that way we have just taken, to make our understanding more simple, we have taken a simple sentence here. Now the same sentence, what happens in the last word, in the last codon, so to say, the last letter got replaced with R. What was T earlier, now it got replaced by, or we are replacing it by R. Then what happens to the sentence? The dog beat the car. So what was cat has become car now. The entire meaning of the word has changed, and following which the meaning of the sentence also changes to some extent. But still the sentence makes sense. There is change in only one word, not the entire sentence. Okay, so accordingly, when we talk about the bases in DNA, the replacement of one base pair will change one amino acid. It will not change the entire polypeptide in cases of point mutation. So here we have it. Point mutations affect one amino acid in the protein, causes no change in the function of the protein, or even if it causes change, there will be some partial change in the functioning. So like this case, you see the dog bit the car. Of course, it has changed the meaning, but it is conveying some meaning. Okay, similar to the previous one. So here one example which we had learned, okay, in the previous lesson, we had previous chapter, we had learned about sickle cell anemia. That is an effect of point mutation. So here we have the example of sickle cell anemia itself I have taken here. So the DNA sequences, the gene coding for hemoglobin structure. This is a part, only a part of it, okay, part where which codes for hemoglobin. Okay, as we had already learned, okay, alpha and beta chains. So beta globin chain, okay, so beta globin chain changes here. This particular sequence of DNA is coding for beta hemoglobin. It will undergo transcription followed by translation. Translation will result, here I have not shown transcription, okay, so DNA sequence will undergo transcription and then translation. Now translation will result into arrangement of the amino acids in this form from the particular DNA sequence shown here: valine, histidine, leucine, threonine, proline, glutamic acid. So this way it forms the polypeptide chain, which is normal, which normally exists in humans. That will give the normal red blood cells. Now what happens here? The pair that is marked here. Okay, the pair that is marked here, some changes occurs at times. Okay, or already it has occurred in human population. It has already occurred in some individuals which are being inherited over the generations. Now, what happens? This particular pair, what was TA, got modified or got substituted. Okay, got substituted in the form of AT. What was TA got substituted as AT. This change we will call as point mutation because it has occurred at only one point. Okay, but the consequence, you see, earlier what CTCGAG was coding for glutamic acid through the RNA. Now due to change in this particular base pair, it is coding for valine. Okay, it is coding for valine. What it was coding for glutamic acid, now it is coding for valine. Resultant, only one amino acid changes, resultant the polypeptide chain. Okay, resultant the polypeptide chain will change to a deformed structure, deformed way, that is, it will not remain biconcave anymore, it will become sickle shaped. Sickle shaped, and the deformity we call as sickle cell. So here we got the example of point mutation. Change in one base pair which was replaced. It changes the structure, the phenotypic expression of the particular gene. Now we will move on to the other type, that is the frame shift mutation. In frame shift mutation, as already mentioned, it involves addition or deletion of one or more bases to a gene which will result in frame shift. Okay, the shifting of the reading frame will shift. This disrupts the reading frame in completely different translation. Like compared to point mutation, in point mutation what happened? The translation was not totally disrupted. Only one amino acid changes. But in case of frame shift mutation, depending upon at what point the mutation is occurring, the total formation of polypeptide might be transformed. Let us see how. Now here the same sequence I've taken here. The dog bit the cat. This is the normal one. Now what can happen here? Like say the the, it remains same. D, the letter D has been deleted. Okay, the letter D got deleted, got removed. So that is in a, but if we say this to be a base sequence, okay, base sequence is made up of the nitrogenous bases, say one pair of bases gets deleted, gets removed totally. When this gets removed, as we are already familiar that the codons, they are read in triplets, in sets of 3. So the reading frame will continue to be in sets of 3, but as D got removed, so the reading frame now shifts. What was earlier, these 3 were taken together, first tree, then next tree, and so on it went. But as D got deleted, now what will happen? The reading frame will shift to OGB here. As I've shown here, OGB takes the second, comes in the form of second triplet. So when if B has gone to the previous codon, it has added up with the previous codon, then what will happen? Accordingly, there will be shift here. So here ITT becomes the third codon. HEC becomes the fourth codon, and then it continues in this manner. So what can we infer from it? That removal of one base or removal of one letter can shift the reading frame. It will shift the reading frame because the during transcription, after transcription, or during transcription, or during translation, always the codons are read in sets of 3. Sets of 3. So irrespective of whatever codon is there, the reading frame will continue in sets of 3. So you can see the total, the meaning, earlier what was conveying a meaning, the dog bit the cat. Now in the second line, it's not conveying any meaning after the. It does not convey any meaning. Totally the polypeptide or the meaning or expression changes. Likewise, like suppose here, this is the normal sequence. Normal sequence, between E and D of the two triplets, one letter is added. Like here, that there is addition of letter T. Letter T, when it gets added, again, as the previous case, the reading frame will continue in sets of 3. So as the new T got added, the reading frame shifts to TDO. So T got added here. So what will happen? The reading frame will become TDO. G will move to the other, GBI. Then accordingly, 3 triplets. So triplets, now here what happens? The base sequence has become longer than the first case, and in the second case here it got shortened. But here, so the reading frame shifts. Okay, earlier it has shifted backward, now it has shifted forward. It has shifted forward. So after the word there is no meaning. It does not convey any meaning. So accordingly, in base sequences, when there is deletion of a base pair or addition of a base pair, there will be total disruption of the polypeptide expression. We will further look into it with the help of DNA sequences. Now, this is the RNA. Okay, here we are talking of RNA now, because mutation can occur at any level here. Okay, so we are talking, if it has occurred in the DNA also, the change will be seen in the RNA. If it occurs in the RNA level, then also the same thing. So let us say this is a normal RNA which was formed out of a DNA after transcription. So it was coding for the first triplet, AUG coded for methionine, AAG coded for lysine, UUU coded for phenylalanine, GGC coded for glycine, and the last 3 codons were coding for stop codon. So this is one complete transcription structural gene part, total, complete. Okay, now deletion frame shift. What happens in deletion frame shift? Here AUG, methionine fine, AAG, lysine fine, UUU was supposed to code for phenylalanine, but what happens? One of the U here goes missing. Okay, it gets deleted. As it gets deleted, the reading frame will shift to UUG. This U went missing. So reading frame will shift to UUG. What was earlier UUU, now it will become UUG. UUG, which will convey a different, or which will go to form a different amino acid if it is there, or maybe none. accordingly, the next amino acid, the fourth codon, so fourth codon will be GCU. GCU. Now this U was a part of the stop codon. Now what happened? The U has entered into the previous amino acid, followed by AA. So there is no stop codon. There is no stop codon now. Now the stop codon, it will not code for any stop codon, AA, and if anything follows, the next one will come here. So what happens? The polypeptide which was supposed to form with methionine, lysine, phenylalanine, glycine and then stop codon. Now what will happen? It will form methionine, lysine fine, because till this point there is no change. But in place of phenylalanine, some other amino acid, next some other amino acid, and finally even the stop codon has got disrupted. So what happens? The entire polypeptide expression will change. Such a mutation is called as deletion frame shift. Insertion frame shift. So insertion frame shift, you see here the same sequences. Okay, this is the normal one. So AUG remains same, but what happened just after guanine of AUG, one new base got inserted, U. One uracil got added here. Okay, it got inserted here. So what was to happen? Actually the reading frame should have gone AAG, but now what will happen? It will read as UAA. This becomes a triplet. This becomes a triplet, followed by GUU. Earlier there was nothing which was read as GUU. Earlier there was nothing which was read as UAA. Then again the next UGG. Okay, so everything shifts here. So you can see here now C will be read with UA. So there is shifting, forward shifting of the basis of the reading frame or the triplet codon. So it will not form the same polypeptide as normally it should have. So what will happen? The resulting polypeptide will be totally deformed, reformed. It will be totally changed from the normal one. Why is it happening? Because a new base has been added in it. This is known as insertion frame shift. Reading frame shifts. That is why it is known as insertion frame shift. But in some cases, what can happen? There may be addition or deletion, but it will not result into frame shifts. Like we can see here. AUG, UUU, these are the same sequences, this normal sequences. But what happened? After AUG, 3 bases were added. Not 1, not 2. 3 bases have been added. If 3 bases are added, then what will happen? These will be read as one triplet codon. So AUG followed by reading of this triplet codon, and the rest will remain same as the original one. So here what you will get? You will get UUU. Okay, that is phenylalanine. So accordingly the reading frame will remain the same. Okay, so this will not result into any frame shifts. It will not result into any frame shifts. Or if this goes missing, okay, here if it goes missing, AAG, okay, that means AAG, this total thing is getting missing. Okay, these triplets are going missing here. If they go missing, means lysine will not be formed, but the rest of the amino acids will be formed. Or if new bases are added, if 3 bases are added together, then that will be read as one triplet codon. So it will not result into any frame shift, but of course the resultant polypeptide, some change will be there in a particular amino acid. Then we come to the effects of mutation. What happens due to mutations? Mutations can be harmful, they can even be helpful, and some may not have any effect at all. So harmful, they can change the protein structure or gene activity in a negative way, like we have seen sickle cell anemia. Okay. Then can be helpful, change protein structure or gene activity in a positive way. In a positive way, like say it increases disease resistance, can create better physiological function also. Sometimes it can be even for basically evolution also now it is said to be Okay, theories have been proposed that evolution is also a result of mutation. So how can we say that mutation is always harmful? Mutation is definitely a harmful uh, phenomenon occurring in nature. Sometimes it can be helpful too. Then some mutations may not have any effect at all. They may not have any effect at all in the sense that, like say a base sequence has changed, okay, like as we all know UUU codes for phenylalanine, suppose, and UUC also codes for phenylalanine. Normally what was existing in the body? UUU. The point mutation occurred at the last code UU. U in place of the last U, C replaced it. So it became UUC. So what will happen? Earlier also it was forming phenylalanine. Even now it will continue to form phenylalanine. So finally what will happen? It will not have any effects. It will not show any effect in the individual even though mutation has occurred. So that was about mutations. Okay, that was all about mutations that we had to learn as per our curriculum. So next day we will move on to another topic. We will move on to the operon systems. So continue listening to the lectures. Keep following the lectures. Thank you.